Hello, everyone. It looks like we are live. Thank you guys so much for joining me on this Wednesday evening for an random open mic. Dr. Dan, aka Creation Myths, just had a really great conversation with Sal Cordova, um, aka Evidence and Reasons, on ENCODE and junk DNA. Really good chat between the two of them. So far, I have Orge and a rational empiricist with me, and the link to join is in the sidebar chat. Welcome, gentlemen. Thank you so much for joining me.
Howdy.
Pleasure to be here.
So, you said it, a rational empiricist, that you had a couple things you wanted to talk about specifically.
Yeah, I guess. Well, you may have seen me and CRISPR in the side chat talking about some topics related to evolutionary development. And I guess to hear your initial thoughts on that, like, are you like how important do you think development is for evolution? I guess to start off with.
I would say it's pretty important, especially to be able to understand it. I particularly love developmental biology. I know that's not everyone's cup of tea, but I like molecular, cellular, developmental biology, and that type of fun stuff. So I am definitely a nerd in that regard.
Okay. Let's see. So I guess. I guess I didn't have any specific questions. I guess recently I've been looking into the different kinds of I guess you could call the different kinds of changes involved in development that could be important for the evolution of morphology over time. Do you have any initial thoughts on that too?
Not really. Hey, interest in atheism.
Oh, hey, Speed.
Hey.
Howdy.
Hello, I'm just muting stuff.
Okay. So
I'm good now. Okay.
Oh.
Yeah. So I captured those papers. There were 4 of them, and I sent them to Dr. Dan. And I do have took a look at them. They're very interesting. All the ones I talked about. I really, really wish people would start linking their papers.
yes, I try to link all my sources in my videos, especially in my debates and all. My recent debate with Neff, I put the sources in the description for that debate, so anyone who wants to go analyze them can do so.
I do get behind because it's a lot of work, because you've got to link. Okay, you got to find the paper, see if you can find a PDF for it. Then you have to have the name of the paper, and then you have to have the actual link so somebody can find it. So it's a lot of work.
Yeah.
I don't know. And doing a lot of stuff today with gene regulation and looking at embryogenesis and gene regulation. And it is incredibly interwoven. And
yes.
Okay, you can have, you got you got a promoter, it's usually a TATA box, TATA, with a lot of thymine and adenine in it. And that is, that's right before the gene. But then behind that, you can have enhancers, and the enhancers can either be repressors or enhancers. And those are called cis-regulatory, and they back up about 60 nucleotides. And then you can have these they have different terminology. They call the far ones enhancers, which are way off. They could be like 1 million nucleotides off, and they fold over. And Cell mentioned one that is actually in another chromosome, and it actually folds over and gives you this, some gene regulation. Well, then you can also have enhancers inside of introns in other genes, and I don't know how this works exactly, but in the gene that is being enhanced, which is, and all of this, I mean, when you start to add it up and you see all the different methods, it looks a little messy to me. It looks a little like things kind of clunked together evolutionarily. Is that a word?
What'd you expect? Speed life is pretty messy.
Yeah, life is pretty messy stuff. But I mean, and then I downloaded 3 books on gene regulation and stuff, and it is an intense study. Of course, I was studying ribosomes last week, and I failed at that. I never get as far as I want to get, you know.
Yeah, me neither.
But it was interesting to watch those two read the same paper, and cells like what he said about the brain, just really, the neurons really hurt my feelings really bad. I think he was suggesting that each individual neuron had individual genetic encoding, every individual neuron, and sort of implying that was how memory. Did you hear that part? Anybody hear that part?
Hear what part?
The part about neurons, where he was kind of claiming that the complexity in the genome, all of that quote unquote junk DNA, had something to do with encoding neurons so that you could find out where the memory is or something. So he said every neuron is different, which is patently false.
Yes.
And he was suggesting, I think, that all of that human complexity had something to do with neurons. And then for onions, he made the same claim that SFT makes, that the reason onions have, 16 or 32 billion nucleotides, the reason for that is to make onions real versatile in variation. Well, garlic doesn't have that luxury. They have a smaller genome, I believe. And onions don't do well going from northern Minnesota down to southern Minnesota. They do better in a colder, climate. So they're iffy, but these huge genomes that they have for like onions and certain amoebas and stuff like that. There was a really good PBS documentary about these gigantic genomes end up being what we would call living fossils, because they don't change very fast. They can't, because they have so many copies of the same genes that if you have a mutation in the gene, the plant ends up doing the same thing, or the animal. And so you end up with a lot of stasis. But then if some of these things lose a tremendous part, amount of their genome, if they shrink down to just one copy, then they start to evolve faster, which was a really kind of an interesting. Has anybody heard of this before, that concept?
I haven't, but it makes sense.
Yeah, it's kind of cool. But so like that lungfish, which is kind of a real throwback. I mean, it has it has a genome, I think that's like, 100 times the size of ours, and it never changes, so it can't mutate because it has so many copies, of the genes, which is pretty interesting. And that's why it doesn't change. So it makes it very static, very stable.
Interesting.
Yeah.
Okay. Specifically, are the regions you're talking about, are they pseudogenes, or have they figured that out yet? Or
I don't know if they're pseudogenes or if they're flat out copies of the same gene that are still functioning, and you just end up with more copies. But they have, it isn't polyploidy. They actually have huge genomes. They've been copied over and over again into the genome. And I would have to look at one and research it more carefully to know exactly what I'm talking about. But I just hit upon this documentary that's making that claim. So, which is worth looking into. There's so much to look into.
Yep. Yeah, and that's the beauty of science. It's a continuing, ongoing process, learning about our world and all of that. That's the beauty of science.
Well,
what are you?
Go ahead.
What are you focusing on, David? In school.
So I'm a biology major.
Okay.
So not quite sure exactly which part of biology I want to actually zero in on, whether it be molecular cell developmental biology. I'm also really interested in ecology and evolutionary biology. So there's a lot of different ways to go in there.
Yeah.
And I'm also really fascinated with neuroscience. So let's just see here. UMBC biology program. I'm just going to see my...
Did you say neuroscience?
Yes, neuroscience.
Yeah, I was going to do a thing on the limbic system today. I put up an icon for it, and then I totally forgot to say anything about it.
Yeah. Yeah. So UMBC biology program has focuses in cell biology, computational biology, developmental biology, and immunology, evolutionary biology, molecular biology. So many great things. I guess the only thing that I'm really not interested in that much is plant biology and computational biology. I just find plants so boring.
But they're so crafty, and I
Yes.
think there's so much evolution out there in plants that we never look at them. You know, we
Yeah.
treat our
100%.
plant buddies badly.
100%. I agree, but I'm just really not into botany and all that type of stuff. Maybe if I actually got a chance to do some types of labs or stuff like that, and my research, maybe I'll change my mind on that.
Well, that's the
Well,
joy of
it's-
going to college, having those opportunities to try that stuff out. You know, that's exactly it.
I mean, it's kind of the same thing with sponges. They don't really move or do that much, so people aren't, or like it's kind of the same thing basically. And I don't know if it's the case with you, David, but like it seems to be that things that don't move or don't do anything interesting, people don't really research that much.
Yeah.
What I love about plants is that they go through, they're doing embryogenesis all of their lives. They continue to do it for their entire lifetime, right? So they not only develop from a seed, but they keep growing new leaves every year and new branches, and so they're embryos constantly. They're always children.
Yep. They are fun in that regard, especially since we can see evolution happen pretty much in real time, and we can manipulate the genes of certain plants by artificial selection and get so many new traits and features, and that's the beauty of GMOs. Gotta love them.
Yeah, David, just a quick request. If Dr. Dan does show up in here, after you ask him your questions, would it be all right if I asked him a question of mine related to this development topic we've been discussing?
Oh, of course. I'm sure Dr. Dan would love to talk about that type of stuff. So he said he's coming on in a few minutes, so I'll just wait for him.
Were you able to look at those books, the chapters, the 2 books I gave you, Andrew? Yeah.
Oh, I thought we weren't supposed to be talking about that publicly.
Oh, we can talk about that we had the books. I paid $400 for the books. Okay, so I want you to know that, and I borrowed you a copy. So that's what we did. Oh. Nothing illegitimate going with paying these guys. And not only that, but like 2 years ago, I gave them $20,000. These Pearson jerks. So I mean, I pay for books. So
okay, sorry, I must have misunderstood, Speed.
No, I just there was just something else that we weren't supposed to talk about. So,
okay.
Anyway, we'll talk about it later. But anyway, did you manage those 2 chapters? There's 2 chapters. There's something called the development toolkit, right? Which the 2 books have a totally different approach to this. The first one, I think, what is it? I got it right here. It gives you. Gilbert and Barisi gives you a lot about gene regulation, whereas the other one, which I forgot the name of also, gives you the development toolkit. They give you like these tools, and one of the things is this heterometry. Is that how you say it? Yeah, all these different ways that these different little tools for development. There's about 20 of them that you'll learn, and you pretty much got a really good idea how an embryo develops. Intercell communication and cell matrices and all of this really incredible stuff. But I thought if we got into those 2 chapters, and I did some PowerPoint presentations that we might be able to explain them in a way. Because when I read that stuff, all of a sudden I was so sold on evolution that it's like a religion for me. Did I say that out loud? Are we
Yes.
live? Are we live?
Yep, yes, we are live.
Oh, it's a religion for me, really. I mean, I worship.
Careful, Speed. You're going to become the next Michael Ruse.
Yeah, I worship this stuff. I mean, it is so clear how things evolved after you look into embryogenesis. That's why, David, I truly hope you go into molecular biology development, embryogenesis.
Yeah.
Devo. The Devo.
Yep, Evo is really fascinating. The thing with Evo is that we can make very specific testable predictions that we should find and what we should never find, specifically as it relates to the fossil record. The further you go back in time, the more quote-unquote primitive fossils should look like, which is exactly what we see with Homo erectus, Neanderthalensis, Floresiensis, etc. And that's why creationists have such a hard time with them.
Yeah. Oh, next time Orge comes into the room, we have to say rocks every 15 minutes.
Rocks.
Otherwise, he leaves. And I've been trying to get him in to do the cold slab thing. We talked this morning about it.
Yeah. Geology bores me to tears.
Yeah. Anyway.
Yeah. I just could not get into geology. The only thing that I like about geology is volcanology and earthquake stuff. That type of stuff is kind of fun.
Oh my God, minerals are the most fascinating things.
I like tectonics.
Minerals, boy, minerals are just they're where it's at. I mean, they're so
minerals.
New religion, minerals. Anyway, I'm too talkative tonight. I've been having some coffee. I'm sorry.
That's okay.
Well, like Speed, you also mentioned stasis earlier, and that kind of got me thinking. Like, you always hear, like, when you go to the fossil record, creationists and scientists they see that some organisms haven't changed that much, at least morphologically, because we know, like, coelacanths, for instance, are changing pretty rapidly. I believe genetically. I may or may not be wrong on that, or but like you always see them. They look and they see this stasis, and they say, "Look, no evolution. Therefore, evolution as a whole is false."
Yeah, in that one group of fish.
Which, of course, is nonsense. Since let's see here, 150 billion base pairs. Not split it. It has 4 species, 40 chromosomes, 50 times larger than that of a human haploid genome. I'm just reading straight from Wikipedia here. And
is that coelacanth?
What was that? Yeah. Okay, yeah. I was looking at the wrong thing. I was looking at this is a plant, Paris japonica. Probably mispronouncing that. 150 billion base pairs. Well, and the cuttlefish, about 43 billion base pairs long. So, really, this is to me. This is not evidence of design.
No.
Because if you're an intelligent designer, simplicity is one of the hallmarks, I guess you would say, of design. So why would you have organisms that are nowhere near as complex as, say, a human, with hundreds and hundreds of times more DNA than a human? Like, why?
Can you put that plant in chat, or a link to that plant that you mentioned?
Yeah, I was just looking up.
That one sounds cool. I want to put that one on my radar.
I think what they're getting at with the complexity thing is that their basic argument is that it couldn't have happened without a designer, which of course is an argument from ignorance, but I think that's kind of the way they see it. Would you guys agree? Yeah.
So yeah, this is the plant that I was reading from.
Okay.
Any interesting things? Try and zoom in.
Paris japonica or japanelka, or is that japonica? Okay, Paris japonica. Cool.
Yeah, there we go. Paris japonica.
Yeah.
Or however it's pronounced. Yep. Species of Japanese plants in the genus Paris in the family Melanocytidae. I'm terrible with Latin, so you'll just have to forgive me. 150 billion base pairs long.
Oh my god, that's a big
an
one.
octopod, and is suspected to have a hybrid of 4 species. 40 chromosomes. Wow. 19 billion more base pairs than the previous record holder, the warbled lungfish. So yeah, it is really fascinating stuff.
Oh, it beat out the lungfish?
The lungfish.
Yeah.
Wow. 133 billion base pairs, the largest known genome of any vertebrate and one of the largest of any organism, along with the Polychaos dubium, I guess I'm pronouncing it right, and 670 billion and 150 billion base pairs. What is this here? A freshwater amoeba. This is really quite interesting. Hopefully Dr. Dan shows up soon. Dr. Dan, I'll be in there soon, I promise. So yeah, hopefully you can educate us a little bit more on this type of stuff. So largest genome size, really, really fascinating.
Yes, dubium. That's a nice one.
Yeah, you would never expect that a fish and a plant would be the
winners.
The largest genome.
Yeah,
it seems very counterintuitive.
It does.
Well, everything is counterintuitive to our little ape brains.
Yeah. Right, exactly.
That's why we have science.
Yep.
So what is the cabbage, the mustard, the broccoli? What is the...
Yeah, it's kind of like... It's kind of like in science and cosmology. You would think that the blue stars are cooler and the red stars are hottest because you kind of associate blue with cold. But in reality, it's the blue stars that are very, very hot and the red stars are the cooler stars. So it's very much counterintuitive sometimes.
I would argue that... Oh, go ahead, Speed.
Oh, brassica. I was trying to think of the name of that cat.
Yeah, brassica.
Okay, yeah. Anyway, my great niece is breeding and studying brassica. So
interesting.
Yeah, I was just gonna say that, like, real quick, that I would argue science is probably the best way we have of, or the best method, I should say, of kind of counteracting the intuition we get from day-to-day experience.
Oh, of course.
Yeah.
Religion certainly. I'm not going to go farther with that, actually.
Yeah. Religion seems to put a block. Well, it was interesting to listen to Sel and Dr. Dan talk and look at the same paper. And at one point they came up with completely 180-degree ideas, right, based on it.
Yeah.
And you got to wonder if Saul was getting that basically from his religious belief. Well, where's Dr. Dan getting his opinion?
Right. Well, we can ask him that when he comes in.
Yeah, if he doesn't have a religious belief, I think maybe I don't know.
Yeah,
maybe from a knowledge base, you know.
Yeah. I think the problem with and I love Sal personally, but I think the problem with people like the logic like Sal and Dr. Tour and all of the others, their religious belief is kind of seemingly informing their scientific belief and not the other way around.
Exactly.
Well, yeah, I guess so. Okay, I want to see, good evidence for evolution. So I'm always looking for that when I read the paper. So I guess that is a bias. And they're always trying to see good evidence for creation. And that is a bias. So I guess we have equal bias. So we're equally biased. So that would explain the 180,
Well, I would kind of argue the opposite. For instance, like when I first learned about all this Evo Devo stuff, for instance, like I was extremely skeptical of it because I heard contrary things all my life. So I looked into it, and now my my mind, of course, has changed, but like it just goes to show how like I might not be the best example, but like it just comes to show how if you just follow the evidence, you're going to change your mind, hopefully.
Yeah.
Right.
When I first started studying embryogenesis or embryology, I was kind of looking for how genes would be a blueprint to build things, right?
Yeah.
I mean, I didn't think that was how it was going to turn out, but kind of in the back of my mind, that's what everybody thinks, that, you know, the genome is like a blueprint. It's going to tell me how to build this structure, that structure. And that isn't anything at all like how it works. And you get into it and you start to see this Rube Goldberg machine of, you know, changing things. And you really start to understand at a deeper level that, you know, it is not a blueprint. It is so far from a blueprint that it's a totally different kind of animal. You know, it's more like complexity or like chaos. You know, chaos theory and fractals, that sort of thing. It's more like fractals.
Yeah.
Yeah, the interesting thing about Dr. Tor, the real irony, is that Dr. Tor admits that intelligent design is not science. He admits on his own website, quote, "I cannot lay the issue at the doorstep of a benevolent creator or even an impersonal, intelligent designer. All I can presently say is that my chemical tools do not permit my assessment of intelligent design," end quote. So pretty much Dr. Tor admits that intelligent design can't really be science. Anyway, Dr. Dan, really good talk tonight with Sal.
Yeah, that was a fun
job.
conversation.
So
I really like that format of having somebody on and just kind of talking for an hour and not having like we're gonna have a formal debate, but like here's a topic, let's just talk. I really like that a lot more than the formal debates. I think that's
I agree.
more informative for the audience.
I sent you links. I sent you links for the 4 papers that you guys showed. So
Thank you.
4 or 5 or whatever.
Yeah.
Yeah, I did. I took the bullet for you guys again.
So Dr. Dan, I find it quite poetic that Maddox had to give you $500.
Oh, that was so
you know,
wonderful.
to his credit, he didn't have to. We all expected some fine print to invalidate whatever I did and or said. But
Well, I fully believe that I embarrassed him out of doing that. I think you should actually share that. You should share that prize with me, Dan.
Should share.
Yeah. And apparently Maddox doesn't seem to understand satire.
Yes, that was actually Speed. Let me say, I thought the weather report was super fun today. I thought your going from the IHI video, the in his image video, and using that as a springboard more broadly into the underlying agenda with education and theocracy. I thought that was a really, really fun conversation. Like I really enjoyed listening to how that went. That was really fun.
Well, I was thinking that we missed today, you know, that we kind of missed, that we missed. Okay, cool. Thank you.
No, that was great.
Did you see the Erica thing with Maddox?
That was the funniest damn thing.
Yeah.
Man.
Yeah. Anyway, one question I had for you is the whole issue of DNA being quote unquote a code that I think was mentioned between you and Sal. Can you just fill me in on what that whole debate even is about?
Yeah, so in terms of its relevance for like evolution and creationism, the argument from creationists is that codes cannot evolve because you need it's an irreducible complexity argument. You need a sender, you need a recipient, you need mutually recognizable symbols, you need an interface between the code and its meaning. The meanings of the characters in the code, whatever form those might take, must be assigned to some real world thing that they correspond to that may or may not be random. So you need all these components that if you're lacking any one of them, you cannot have that communication. Therefore, codes cannot evolve. It's an irreducible complexity argument. So creationists, and I think Maddox falls into this category, make the argument because if you acknowledge DNA as a code, then therefore you're acknowledging that the system of gene expression in DNA could not have evolved. Now, the problem with that is that they don't do the next step, right? So you go, okay, DNA is a code. And they say, okay, well, therefore it couldn't have evolved because all codes come from minds. And you go, well, all human-made codes come from human minds, but we also have evolutionary processes, and they can make the genetic code. And then if you go back and watch the thing with Maddox from the other night, there's no next step. There's no like making the argument. It's like that comic where it's two guys at a chalkboard and it's like math, question mark, and it says, and then a miracle occurred. And it's like, you need to clarify this step.
That's
and what creationists like to do with the code thing is they'll say DNA is a code, therefore creation. And it's like, well, there's a few dots in between there that you need to connect, and they can't do that. So that's why the code argument comes up.
So basically, it's kind of like it's a code, therefore there's a coder. Kind of like there are scientific laws, therefore there must be a lawgiver. Kind of like that type of stuff.
Yeah,
I think that's one way to frame it. Yeah.
Demeter's papers, he talks about like a very, very rudimentary ribosome, with an actual anticodon, and then he talks about how basically guanine and cytosine were highly more abundant, I think. So it was GC rich, and GC rich structures ended up being more stable. And then they start looking at like the minimal set of amino acids. So you got like 6 amino acids. So you only need like 2 nucleotides to code some of that stuff. And you can kind of see where the tRNAs started to statistically form. And we don't know the steps yet, but you could start to imagine the steps between the two things. And you just got to look. But if you don't look, you're not going to find them. If you say a miracle occurred, you're done, right? You go home.
Jimmy,
Jimmy,
know the answer. Oh, go ahead.
Okay. To me, intelligent design and religion is simply an excuse to stop thinking. Okay, we have these difficult problems in biology. How do we get light, for example? Well, we don't have to think about that anymore because God did it. It's basically a cop-out.
And I saw this amazing thing in a book about bones and cartilage. There's let's see, I think there were some genes that go back to fish cartilage. Like fish have more kinds of cartilage than mammals do for some reason, and they related it to some disease. So here was a case where looking at the evolution can give us clues for a chronic degenerative disease in humans. And I have to find that piece in the book again and try to understand it more. But that was like, okay, when they ask, what does evolution do for medicine? Well, there's an example right there. Looking at fish cartilage and the genes for fish cartilage, we were able to see where a mutation in humans caused a, pathology. And we're looking back in time to see how that occurred. Anyway.
Now, a creationist would say, well, that doesn't mean they share a common ancestry. It just means they were created with the same mechanism, so you could study one to learn about the other. And further, they were designed that way. God created it that way so that humans could learn about creation by studying other organisms. That's the excuse you get. I'm not saying it's a good argument. Just saying that's the argument.
So, Dr. Dan, Scott Duke in the Cyber chat asked, when appropriate, please have Dr. Dan reflect on his dialogue with Dr. James Carter. So I guess if you have any thoughts on your... I thought it was a great discussion.
That was super fun. Like Sal tonight, Carter on Monday, I really like talking to creationists who are in whatever field we're talking about. So Carter was super fun. I mean, he's nice, and it was very pleasant, cordial conversation. And he suggested the topic of the Lenski long-term evolution experiment. And the thing for me that surprised me, because creationists have been talking about this experiment for a long time, because there are a few parts of it that creationists can kind of glom onto and say, aha. like the fact that the genomes have reduced in size over time, or that you do see a net loss of genes in these E. coli. Now, the reason for that is not what creationists say. It's not genetic entropy. It's
Right.
that you're selecting for a hyper-specific environment for 70,000 generations. You're going to optimize for that environment. And
Correct.
that includes inactivating and getting rid of genes that are not useful in that environment. So that explains that. The thing that surprised me, I think, was that all of the sources that Dr. Carter brought were specifically from creationists. So he cited the Scott Minnick paper.
Yeah.
That paper is a legit paper. It's in a legit peer-reviewed journal. But that's the only one. I think there was a Behe one that was also peer-reviewed. But then there was the Casey Luskin stuff. There was the Sanford stuff. It's all creationist sources. And they do not do a good job of just describing the basics of what happened in that experiment. And, Behe saying there was this big die-off in the citrate, the CIT plus line. Well, they had a very high mortality rate when you changed environments to something that was only citrate.
Right.
Obviously, they didn't adapt to that environment. You take something and adapt it to this one thing, and then you put it in something else, it's going to be worse off. In
Right, of course.
the original environment,
It's like being surprised that an Arctic fox is not going to be a very good fit in a forest.
Mhm.
So
that was the big surprising thing for me, was that I would expect somebody to, like, my and I get this, actually, they're live right now over in Evolution Hour. RJ Downard his thing is source methods, right? Go to the original source. If I'm going to talk about the Linsky long-term evolution experiment, I'm going to get some papers I'm going to get the papers on the CIT plus trait from those people.
Right, exactly.
They're the ones that have done the work to show what happened, how it happened, when it happened, right? I'm not going to rely on the creationists explaining what those papers say. Just go to those papers themselves.
Exactly.
That was surprising to me.
Right, and I must say, I absolutely love RJ Downard. He is a really good guy.
Everyone should go. They're live right now. I was watching it a little bit over for Evolution Hour. Him and I think it's Jack. I think Jackson's on there now. Yep, it's Jackson Wheat is on there now. They're great. That's a fun show.
Yeah. He doesn't get enough people watching. I don't think we need to. I keep forgetting.
If you're watching this, go watch Evolution Hour with Art. Sorry, David.
But anyways, Dr. Carter seemed to agree that irreducible complexity has been falsified, if I recall correctly, one of his slides.
I don't know if I would go that far, because you get a lot of flexibility in what that means. You get the original Behe definition, which was very limited. You get the revised Behe definition from like 2002, I want to say. And that definition is very specific. He actually says it's a trait where any step in the development of that trait, the evolutionary development of that trait, would be unselectable, which, that is a very broad criteria. And let me tell you, there are a lot of traits that I can point to where some intermediate steps are unselectable. Like, that is not a hard standard to meet. And so I think it's perfectly clear that with minimal work, you can find a bunch of examples of observed traits that meet the criteria, the canonical Behe criteria for irreducible complexity. The question is, go ahead.
Yeah, my personal favorite being the HIV virus, which we know exactly how it evolved. Create the phylogeny sporids from SIV.
We got it.
And again, I think it's stuff like this that really compels me for evolution. If evolution were false, then why does it work so well in explaining basic biology and virology and stuff like that?
Yeah. And going back a minute to something Speed said in terms of just like predictions, like putting aside kind of large scale evolutionary predictions, we can look at very small, very specific things. Like there's some experiments with antibiotic resistance where you take a specific type of bacteria, you expose it to a high concentration of a specific amino acid, and you watch the pathway of mutations that lead to robust resistance to that antibiotic. And what we've seen with some of these experiments is you do it in a lab and you say, okay, here are the genotypes we expect, and here's the sequence of mutations that we expect, where at each step in the pathway, you get an increase in resistance. And in at least one case of that, we've done that kind of experiment. And then a couple of years later, the exact strain that they found in the lab appeared clinically in a hospital. Now, the benefit there is you could go, aha, we've seen this before. We know how to kill it. This is very specific. We can do the experimental evolution in a lab and say, this is what we expect to happen in the real world. And that's exactly what has happened. Like, if evolution is wrong, now creationists will say that's just microevolution, right? Of course, populations have variation and blah, 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 right? That's what they'll say. But there's no boundary line between those principles and those processes and universal common ancestry.
Right, exactly.
So, like, once you open that barn door, every horse is leaving the barn. You can't stop me from making the case that because we can show how bacteria will evolve in that situation, you can't stop me from showing how eukaryotes and prokaryotes share common ancestry, because it's the same processes and it's the same principles.
You basically have to falsify genetics at that point.
Really, yeah, because I mean, creationists. I mean, this is something like Jensen does. Is he says you have common ancestry mostly at the family level. You're like, okay, here's all the sequence similarities without any of the labels. Draw the line at family and show me where the common ancestry stops. Can't do it. It's you can't do it. It's just a constant. It's a nested hierarchy where things that share more recent common ancestry are more similar. But at no point does it just stop. And the great thing is, we have a counterpoint to that to test it against, because we have viruses, and viruses don't share universal common ancestry. There are probably a dozen or more different origins for viruses. And we know that because you try to do the phylogenetics for viruses and you can't. They don't even have the same genes. So you get one clade of viruses and, nested subgroups. You get another clade of viruses and all the nested subgroups, but there's not even a comparison to be made between the two. They don't share common ancestry. In many cases, their genomes aren't even the same material. You got double-stranded DNA, single-stranded DNA, two different types of single-stranded RNA, double-stranded RNA, retroviruses, right? Like, there's no one phylogeny for all that stuff because they don't share universal common ancestry. The fact that you could take any cellular organisms and go backwards and determine their relative ancestry to each other, like that is super, super positive evidence that they share universal common ancestry. It's like that's case closed, because again, we have a counterpoint. We have a null hypothesis to test it against. If they don't share common ancestry, we know what that looks like because we have viruses. Cells have common ancestry.
Yeah, and it's kind of like the RN-ROS phylogeny challenge. Where do you draw the line?
That's exactly what it is, yeah.
If you draw it at the family level, then that means humans and the great apes are the same kind because we're in the same family. Oh, but that doesn't count. Creationists don't like that. And here's the thing.
And Dave, if I could just say, Jensen says that doesn't count. He doesn't give a reason. In Replacing Darwin, he literally just says, obviously humans are different and we don't share common ancestry with the other great apes. For everything else, it's at the family level. Like, he just says there's an exception for humans and he doesn't give a reason.
Right, and here's the question that I have for creationists, and I would love to ask Dr. Jensen this: if every other species had the ability to eventually radiate out and speciate, why can't humans?
That's a good question.
That's a good question.
Yep,
I would love to ask that.
Yep. So,
and
of course they're going to probably respond by saying, well, because humans have low genetic diversity compared to the other organisms. That's pretty much what I predict that they will respond. But that's not how
I just rolled my eyes so hard I saw my brain.
What was that?
I said I just rolled my eyes so hard I saw my brain.
but that's pretty much going to be the typical response.
No, that you're right. You're right. But like, that's not how population genetics works. That's not how it works at all.
Exactly, because you have different, of course, methods of speciation. I think the most common is allopatric speciation, where you have the geographical barrier between populations.
Yeah, there's some interesting cases with what is it called, parapatric, where like Drosophila from Northern California to Southern California, they're doing studies and they sort of grade down, into differences, and there are some plants that do that too. But on the Lenski experiment, I always like to challenge the creationists that if they think that those bacteria have become weak from genetic entropy, go ahead and drink 3 flasks of them. Go and ask Lindsay if he'll give you a little
And here's something, especially with the
That's
current.
that's E. coli. That's it's what is it? Is it K12? I used to work with the same strain actually. It's non-pathogenic. You could drink as much of it as you want. It's not going to hurt you.
Yeah, but after 60,000 generations, would you count on that?
They'd actually probably get outcompeted by the E. coli already in your gut.
Yeah.
And most of them wouldn't survive your stomach anyway because the pH is low too.
Has anybody thought about extending that to a multi-environment test? Are they doing that?
There's tons of spin-offs. Yeah. They do. The famous one is the citrate only that they published last year, but they've done a ton of other ones where basically Lenski has, he's had hundreds of grad students over the years, and many of them will take a lineage of his bacteria with them when they go off and start their own labs. And they will do follow-up work on those in different environments. So it's like if you adapt hardcore for this environment, how do you how does your evolutionary history then limit your future adaptation to a different environment?
And are they sequencing every generation?
It's I know they have the frozen stocks from, I want to say, I forget if it's 1,000 or 2,000 or 5,000. They have the frozen stocks for every couple of thousand at least.
Okay.
And then I don't think they're sequencing everyone, but they do it regularly. They're checking on them. Yeah.
It's going to take 100 years to analyze that data.
It's so cool. I love that experiment so much.
I think the Lindsay's long-term experiment is probably one of my favorite experiments of all time.
It's beautiful. It's absolutely beautiful.
And but it's still E. coli after 40 million generations, therefore evolution's false.
No fishermen have crawled out of that Lenski experiment.
What is it? Microbes to man? Yeah.
Particles to people. Good as zoo to you.
Zoo to you is a good one. Zoo to you is a good one because I like zoo to you because it misunderstands how everything works. Not just
actually it
does it like just
it's goo to you by zoo.
Oh, okay, it's goo to you, but it still has zoo in the middle, and there's nothing extant that evolved from anything else extant. So
1, 2, 3, 4, 5, 6. I can get a license plate, a goo to you license plate. Did I do it?
So, if it's okay with you, David, since we're talking about lineages and stuff like that, I think this might be a good segue. If I had some related questions for you, Dr. Dan, if it's okay with
please.
you guys.
I will say it's 11, so in like 10 minutes I'm going to go to bed. I can't stay up that late, but yeah, by all means, go ahead if that's okay with you, David and Speed.
Oh yeah, go ahead. And as soon as he's finished answering that question, I do have one. I guess last question for you.
Yeah, totally.
Okay. So I guess before we've talked about the methods that are used to compare different phylogenetic trees, and I was hoping you could give me, I guess just a basic rundown of the difference between, say, parsimony and or like a basic rundown of kind of like how likelihood analysis works, because I'm having trouble wrapping my head around it.
Yeah. Oh, it's so cool. So this is literally one of my classes every summer is we just talk about these methods a little bit. So parsimony so what we're talking about is phylogenetic methods, right? You have a number of organisms, they have characteristics, or you could do sequences. And what you want to do is figure out the relationship pattern. Parsimony is the idea that you want to minimize the number of changes. So like if you have AAAA, right? You have AAAA, and then in base 3, it becomes AATA for 2 out of your 4 sequences, right? And then one of them is AATC, right? Just make something up, whatever,
Okay.
right? Now, what you want to do is minimize the number of changes. So you could say you mutated an A over here, and then you mutated 2 A's over here. And that requires 1, 2, 3 changes. But it makes more sense, it's more parsimonious to say, well, one of the A's mutated upstream, right? And then within that lineage, another A subsequently mutated. And that's how you get your pattern. So now you only have 2 changes in your sequence instead of 3. So what you're doing is you are trying to figure out the pattern, the sequence of changes that occurred between the common ancestor and your extant groups, and the relationship among those groups that minimizes the number of changes required. So the number of changes in either DNA sequence or amino acid sequence or phenotype or whatever, right? So that's parsimony. It's just do it with the smallest number of changes. Does that make sense?
Yeah.
Yeah.
And does a similar thing apply to say likelihood and Bayesian inference, or is it completely different?
That's a different thing. So that stuff is much more sophisticated. So when you're getting into like maximum likelihood methods and Bayesian methods, and Bayesian statistics are, so like the statistics that everyone knows and loves or hates, depending on how much time you spend with them, are Fisherian statistics. So like when you're calculating confidence intervals and p-values, that's Fisherian statistics. What you're doing with Bayesian statistics is you're kind of flipping it around, right? So like with basically what you're doing is you're setting your priors, it's called, is kind of your conditions. And people who will look down their noses on these techniques will call them your assumptions. And you call them whatever you want. It's your preconditions. And you're saying, okay, for example, with evolution, you'll say A mutates to T at a certain frequency. A mutates to G at a different frequency. A mutates to C at a different frequency. C mutates back to A at a certain frequency. T mutates to A. Like you set things like your mutation rates and your population size and the growth model, right? So you're setting your genetic and your demographic conditions for your population. And then you put into the program, because this is all done electronically, you can't do this stuff by hand.
Right.
Put into the program your alignment. So you have your gene sequences and they're aligned, right, as best you can. And you can do that automated, but like you got to check it by hand and do it by hand if you want to be really good about it. And what the program will do is it will take your sequences. Oh, you know what? My dog is barking. Sorry, I need to just open the door before he barks more. Just a second. Sorry, everybody. Hang on. Everybody, come on in. All right. I apologize. My dog wanted to come in here. He's now just hanging out on the couch back there, but that's sorry about that. So what you're going to do is the program is going to look at those sequences and say, okay, given the parameters that you have provided, your priors, right? Like your base specific mutation rates. Say we know that like C mutates to T really fast. We know that happens all the time. And T mutates to C really slowly, right? So if you have a C to T mutation or several, the program will say, okay, that happens all the time. So we're not going to assign like a high score for homology to those changes. But if you have a specific T mutates to a C, well, we know that's really rare. So if we see the same T to C change in multiple sequences compared to all the rest, then we're going to say that's probably common ancestry that caused that. So based on the conditions that you put in, the program will build many, many trees and then compare them. And then it will pick the best among those and will then mess with those trees. It will perturb them, right? It'll mutate them essentially. It's basically doing selection. It'll mutate them and generate variants of those trees. And then it'll pick the best of those, the ones that score the best according to your parameters. And it'll do this iteratively, 1,000, 10,000 times. And at the end of it, you get a tree that adheres to the conditions that you have provided for the program. And we're not just guessing about those parameters. We actually can determine them experimentally, like, for example, base-specific mutation rates. So there's always something called a substitution matrix, which is at what rate does each base mutate into every other base. And there's lots of different kind of theories and ideas about how best to model that. And like when I was in graduate school, part of my research, part of the work I did was actually determining those rates so that my colleagues could then use more realistic models when they're doing viral phylogenetics. So you experimentally determine the parameters and then use the parameters to say, okay, tell me what the relationships are. Those are the more sophisticated Bayesian or maximum likelihood techniques. Very briefly, it's a lot more complicated than that, but yeah.
Right, and just to clarify something, when you say perturb them, like they're perturbing them based on the parameters, or could you clarify that?
They're some ways of doing this actually change them somewhat randomly. You just kind of break them, and you'd be like, well, we have this branch connected, but what if we connect this one to this one over here and see if that gets us a lower score, right? It's you randomly generate 10,000 trees. You're just randomly connecting your sequences and seeing which variants of those trees are the best. Like the program that I'm familiar with, it does 10,000. I think it's 10,000 and then picks 4. And then of those 4, it generates 10,000 more. And it does that iteratively, like 10,000 times. It takes forever. You have to use like a really intense computer, and it will literally run for like 3 to 6 weeks at a time doing the calculations. It takes forever.
So it's the trees that maximize the probability of the characters giving your parameters, basically.
Exactly.
Okay,
Sure.
that makes
What's the most realistic sequence of events given what we know about the population size, the population growth rate, the mutation rates between all the different bases, right? Given those parameters and these 1,000 sequences, what's the most realistic pattern of common descent for all that? Yeah.
Right. Okay. Thanks. That makes sense.
Right. What is that program name? The program name?
The one, let me think. Hang on. It's called BEAST. It's BEAST, and it stands for something. It's Bayesian something or other. Sequence something. Yeah, it's BEAST. BEAST.
Yeah. Cool.
Is the one that that's what I don't even know if that still exists anymore, honestly. That's the one we used when I was in graduate school. But again, I was actually upstream of that.
Yeah, it looks like there's a BEAST 2 now. Bayesian evolutionary analysis by sampling trees.
That's the one. That's the one.
Yep. And as Call Me Emo pointed out, all phylogenies are hypotheses on how organisms are related. They're not meant to be taken as 100% represented from actual evolutionary lineages, but rather as an approximation of them. And this is absolutely correct. And again, this is how we make if scanning for truth wants testable predictions evolutionists make. Well, there we have it. Those are phylogenies. Yes. And all the time, we're changing the phylogenetic trees because we see, okay, this isn't exactly the way we expected it to be. Therefore, this aspect is kind of overturned, and we kind of have to redraw the lines. So
that's 100% right. We get new data, we reevaluate our tree, we learn something about, for example, the population size for whatever group you're looking at, or with the mutation rates. One thing that my lab did, I wasn't involved in this directly, but again, I was upstream of it, is one of this is getting really into the weeds, so I apologize. I'll do this fast. But there was the mutation rates, the normal thing that people do is called the general time reversible model, where if A mutates to G, you assume G mutates to A at the same rate. The forward and backwards are the same. It's reversible. But that's not accurate. We know that's not accurate. We know that's wrong. And so some of my colleagues developed a more sophisticated model called the Unrest Model, which stood for unrestricted, because it meant that all the rates, the forward and the backward, could all be unique rates. And that's going to be more realistic because we know the rates are not symmetrical. So like that's when you incorporate those new data and use the new version of your model, whatever it is, you're going to get a new phylogeny that should be more representative of reality, right? That's the idea.
And the methods will be the same for both time reversible and non-time reversible models, yes?
In general, yeah, but you're going to get different answers because of the differences in terms of what the rates you expect to see are. Yeah, but the techniques are basically the same. Yeah,
the basic method.
Yep. Mm-hmm.
Okay.
All All right, Neff, did you have anything? Because I am going to go to bed.
Yeah,
soon.
yeah. This is my last question, and this is a great. I guess this will be a good way to wrap everything up. My question related to human evolution. Why is it that like we have Aboriginal Australians who've been there for 50,000 years, separated geographically? Why isn't it that they ended up speciating from the main human groups? How long would it actually have taken for us to have speciated?
I mean, we're all really similar in the grand scheme for something with the generation time of 20 years and, a low mutation rate. It would take a long time for humans to diverge enough to be a different species. And we just haven't been apart for that long. We only left Africa 60 to 80,000 years ago. And that's just not a lot of time for something with relatively small population sizes, slow mutation rates, big multicellular, long generation times, right? That's just not a lot of time. If you could, put, double humanity and put the new half on Mars and then stay apart for 500,000 years, I bet you would get two different species. But
yeah, but
I know, I mean, we're all so similar. I mean,
in the past, we had, of course, quite a bit different human species that seemed to speciate rather quickly back then. So at least that's what I'm making.
Well, the reason for that is you had essentially radiation events, right? So you had small populations in different parts of Africa, and then they radiated from each other, and you end up. I mean, you're talking in the millions of years ago, and there were at least two instances of out of Africa migrations prior to Homo sapiens leaving, right? So you have these radiation events where you have prolonged periods, and we're talking now in the millions of years of isolation from each other in different environments.
Right, like the Neanderthals evolving in the more Arctic
in the northern, right? So that's going to be, I mean, because that would have come from. Oh, when did that, Mike? When did that happen? When did Heidelbergensis? I want to say that was one. I don't know. I don't want to say a number because I don't know what it was.
I
but
think
it was in the millions of years ago, right?
Yeah, like a few 100,000 years ago, somewhere around then.
The migration outwards of Heidelbergensis, would it have been in the hundreds of thousands? I think it was in the millions of years. I want to say millions, but I could be wrong.
I might be thinking about when Neanderthals and Homo sapiens split genetically from Heidelbergensis.
So it looks like 2 million years ago. But I guess this kind of to the whole idea from Dr. Sanford's the waiting time problem, which. But I know there's a lot of now the waiting time problem actually refutes young Earth creationism, especially as it relates to the Noachian deluge. So if Sanford is right, young Earth creationism is wrong. That's all there is to it. But
not the first time. Not the first time, because genetic entropy, as you very astutely have pointed out, refutes created heterozygosity. It's one or the other. It can't be both.
Right.
Right.
Hold on, I'm calling you. When finding an arthropod with a mammalian-style inner ear system and/or vertebrate styles challenge or falsify phylogeny.
Wait, what was that? Can you find an arthropod with a mammalian-style inner ear system and/or vertebrate-style eyes? I mean, finding weird stuff like that is always going to be a challenge for phylogenetics, certainly. But what matters is the underlying genetics of the structure and the evolutionary origins. So, for example, we know like vertebrate eyes and arthropod eyes share common ancestry, not as eyes, but the proteins involved, the opsins and the crystallins, share common ancestry as serpentine proteins, G protein coupled receptors, and heat shock proteins. We know that. So if you were to find organism X with an eye that resembles the eye of organism Y, okay, they're built from the same things. So we actually have an example of that, right? What happens if you were to find an eye that's like a vertebrate eye, but it's in an invertebrate that's almost identical? Okay, that exists in mollusks, mollusk eyes, right? Cephalopods. Right. That exact thing that was in that hypothetical, that exists. But we know that it's two distinct evolutionary pathways, and we know what genes are responsible in each case. And And it's actually not exactly the same genes because there's two types of opsins. And in deuterostomes and protostomes, they kind of switch jobs. So we can figure that stuff out. And I'll give you one more example that's really interesting. When you sometimes find stuff that's weird like that, and you get really interesting, you learn stuff from it, is nudibranchs, which are gastropods. They're relatives of snails and slugs. They're brightly colored, which means don't mess with me. I will mess you up if you try to eat me, because they have stingers in their backs.
Yeah.
If you want to Google image nudibranchs, they're super cool, but they're all really brightly colored. And they have stingers in their backs. And when scientists first looked at their stingers, they're really confused because they look like jellyfish stingers. They look like cnidarian stingers. And cnidarians are not closely related to mollusks. Cnidarians are basal eumetazoans, right? So they're diploblast, they have radial symmetry, they're not triploblast, they don't have bilateral symmetry, and mollusks are very sophisticated coelomate bilaterians. So this was very confusing. Turns out the nudibranchs eat small cnidarians and steal the stingers and then move the stingers from their intestines into their backs. And that's where they were coming from.
Yeah.
Nice.
Yeah.
Yeah.
It's so cool. So you find weird stuff like that, but you got to look into like what the explanation is. And you always learn something cool when that happens.
I think they featured one of those in one of those Deb books I gave you, Andrew.
Yep.
Okay.
Yeah, but anyway, with genetic entropy, Dr. Dana, what I'm thinking, and Justin Walker Fish that he's in for this, is to kind of host a roundtable, kind of an atheist book club, and call it chat and go through it chapter by chapter. So if anyone's interested, let me know.
Oh, that book is so bad. I'm gonna have my blood pressure will be so high. I just don't call it an atheist. Don't call it an atheist book club though. Call it a biology book club.
Yeah, science book club.
Yeah, it's a science book club.
Yeah. By the way,
I missed David.
BBC.
Yeah. By the way, there's a really interesting book. I'm not sure if you've heard of it, called "Evolving: The Human Effect and Why It Matters" by Daniel J. Fairbanks.
I don't know that one. I'm going to look that up, and then I'm also going to call it a night, everybody. Thank you for the book rec. And Speed again. Listening to the weather report today was super fun. So thank you. That was really enjoyable.
I still love how Maddox mistook Gutsick Gibbons' comment as being legitimate.
Well, he did the same thing the other night with another joke. He totally missed.
Right. Bye, everybody.
Yeah. All right. Bye, Dr. Dan.
Dr. Dan, thanks.
David, what book are you? Do you want to do a book club on?
Genetic entropy.
Oh my God, yeah, because I have so much trouble reading that. I would love to go through it with somebody. So if you want to include me in that, I'd love to be a part of it.
Yeah, sure. So that means we all got to read it and kind of set a deadline for getting it chapter by chapter.
Yeah, when I try to read it all at once or something, my head wants to explode, but I can discipline myself.
Yep. All right. Well, thank you guys so much for coming in.
Thank you.
It was a great time with everyone. And don't forget hit like, don't forget hit subscribe. And I will see you next time.
Cool.